Welcome to Anatomy and Physiology Part 2. Today, we're diving into cell physiology and tissues. Let's make it count. The cell membrane, also called the phospholipid bilayer, is made up of two layers of phospholipids. The hydrophilic, or water-loving, heads face outward toward the watery environments inside and outside the cell. Meanwhile, the hydrophobic, or water-fearing tails point inward, away from water. This unique arrangement makes the membrane selectively permeable, meaning it only lets certain substances pass through while keeping others out. The membrane also contains proteins, cholesterol, and carbohydrates, which help with structure, communication, and transport. Now, let's compare the fluids. Extracellular fluid is found outside the cell, while intracellular fluid is inside. They have different compositions, which is important for cell function. Hydrostatic pressure is the force of a fluid pushing against a surface, like blood against vessel walls. Osmotic pressure, on the other hand, is the pull created by solutes drawing water across a membrane. Diffusion is the movement of particles from high to low concentration, while osmosis is the movement of water from an area of low solute concentration to high solute concentration. Both move down their concentration gradients, but osmosis is all about water. And that's your quick review of the cell membrane and its amazing functions. Let's keep going. Selectively permeable. A selectively permeable membrane allows certain molecules or ions to pass through it by means of active or passive transport while restricting others. Hydrophilic versus hydrophobic. Hydrophilic, attracted to water, typically polar or charged molecules. Hydrophobic, repelled by water, typically nonpolar molecules. Extracellular versus intracellular fluid. Extracellular fluid outside cells includes interstitial fluid and plasma, contains nutrients, ions, and waste products. Intracellular fluid within cells, rich in potassium and proteins, and maintains cellular processes. Hydrostatic pressure versus osmotic pressure. Hydrostatic pressure, the pressure exerted by a fluid at equilibrium due to gravity. It pushes fluid out of capillaries. Osmotic pressure, the pressure required to prevent the flow of water across a semi-permeable membrane. It pulls fluid into capillaries. Diffusion versus osmosis. Diffusion, movement of particles from high to low concentration. Osmosis, movement of water across a semi-permeable membrane from low to high solute concentration. Simple diffusion, facilitated diffusion and active transport. Simple diffusion, passive movement of molecules through the membrane without assistance. Facilitated diffusion, passive transport of molecules via membrane proteins. Active transport, movement of molecules against their concentration gradient using energy, ATP. I bet you didn't know just how much osmosis can affect your cells. Welcome back to part 2 of our Anatomy and Physiology series, where we dive into the fascinating world of cell physiology and tissues. Today, we're exploring how different solutions impact cells and why it matters for your health. In an isotonic solution, there's no net movement of water, so the cell, well, it just stays the same. But in a hypotonic solution, water enters the cell, and the cell may swell. On the other hand, in a hypertonic solution, water exits the cell, and the cell may shrink. Enzymes and reaction rates are honestly pretty fascinating. Enzymes catalyze biochemical reactions, increasing reaction rates by quite a bit. Several factors like temperature, pH, enzyme concentration, and substrate concentration all affect these rates. Now let's talk about enzyme-substrate binding. Enzymes bind substrates at the active site, forming an enzyme-substrate complex that actually helps facilitate the reaction. And you know, the importance of vitamins shouldn't be overlooked. Vitamins often act as coenzymes or cofactors, assisting enzymes in catalyzing reactions. Amylase is a key player in carbohydrate breakdown. Its main function is to break down starches into simpler sugars like maltose and glucose during digestion. Speaking of enzymes, let's look at their composition and how temperature affects them. Enzymes are primarily proteins, and extreme temperatures can denature them, altering their structure and function. And finally, denaturation. This is the process by which proteins lose their structure and function due to external stressors like heat or changes in pH. It's a pretty big deal for how enzymes work. Phagocytosis, endocytosis, and exocytosis. Phagocytosis is the engulfing of large particles or cells by a cell. Endocytosis is the process by which cells internalize molecules by engulfing them. Exocytosis, on the other hand, is the process by which cells expel materials in vesicles. Organic compounds are molecules containing carbon and are found in living organisms. Some examples include carbohydrates, proteins, lipids, and nucleic acids. Now, let's talk about the importance of DNA and RNA. DNA stores genetic information and instructions for protein synthesis, while RNA translates genetic information from DNA into proteins and performs various roles in gene expression. The resting membrane potential of a neuron is negative due to the differential distribution of ions, particularly the efflux of potassium ions through leak channels. Potassium leak channels allow potassium to flow out of the cell, contributing to the negative charge inside. The sodium-potassium pump actively transports 3 sodium ions out and 2 potassium ions into the cell using ATP, maintaining concentration gradients and the resting membrane potential. Factors contributing to the resting membrane potential include ion concentration gradients, membrane permeability, and active transport, for example, the sodium-potassium pump. Let's walk through the steps of an action potential. First, during depolarization, sodium channels open and, the membrane becomes more positive. Then comes repolarization, where potassium channels open and the membrane returns to a negative state. After that, hyperpolarization occurs as potassium channels stay open just a bit longer, making the membrane potential even more negative than its resting state. Now, let's talk about the importance of the myelin sheath. Its main function is to insulate axons, which in turn increases the speed of nerve impulse conduction. Next up is saltatory conduction. This refers to the jumping of action potentials between nodes of Ranvier in myelinated axons, which really speeds up signal transmission. Now, let's discuss absolute and relative refractory periods. During the absolute refractory period, no new action potential can be initiated no matter what. But during the relative refractory period, a stronger than normal stimulus can actually initiate an action potential. Now let's compare EPSPs and IPSPs. EPSPs, or excitatory postsynaptic potentials, depolarize the membrane and increase the likelihood of an action potential. On the other hand, IPSPs, or inhibitory postsynaptic potentials, hyperpolarize the membrane, decreasing the likelihood of an action potential. So, let's talk a bit about the structure and function of a nerve cell. The structure is made up of dendrites, the cell body, an axon, and synaptic terminals. As for its function, it transmits electrical impulses and communicates with other cells. Now, where exactly do action potentials start in neurons? That would be the axon hillock, which is the initial segment of the axon where action potentials are generated. And moving on, there are 4 major tissue types. Epithelial tissue covers surfaces, lines cavities, and forms glands. Connective tissue supports, binds, and protects other tissues. Muscular tissue is responsible for movement through contraction. And finally, nervous tissue transmits electrical signals for communication. There are 6 main types of epithelia. First, simple squamous. These are thin, flat cells found in places like the alveoli and blood vessels. Next, simple cuboidal, which are cube-shaped cells found in glands and kidney tubules. Then there's simple columnar, made up of tall cells you'll find in the digestive tract. Stratified squamous consists of multiple layers and is found in the skin and mouth. Stratified cuboidal, also multiple layers, is found in sweat glands. And lastly, stratified columnar, which has multiple layers and is located in the male urethra. Special types of epithelia include pseudostratified epithelium, which appears layered but isn't truly stratified and is found in the respiratory tract. There's also transitional epithelium, which stretches and is found in the urinary bladder. Now, let's talk about the basement membrane. It's a thin layer situated between epithelial and connective tissues, providing essential support and anchoring. Let's look at the types of connective tissue. Dense connective tissue includes tendons and ligaments. Loose connective tissue includes areolar and adipose tissue. Fluid connective tissue includes blood and lymph. And supportive connective tissue includes bone and cartilage. Cartilage healing is a slow process because cartilage is avascular, meaning it lacks a blood supply. This really slows down nutrient delivery and the repair processes. Now, moving on to more types of connective tissue. Elastic tissue is found in the walls of arteries. Areolar tissue is found under the skin and around organs, and adipose tissue stores fat, also found under the skin and around organs. So let's talk about the cells in connective tissue. Fibroblasts, for instance, produce fibers and ground substance. Chondroblasts, on the other hand, form cartilage. Then there are macrophages, which, engulf pathogens. And finally, mast cells release histamine in response to allergens. Now, blood as connective tissue is actually pretty fascinating. Its main functions are to transport nutrients, gases, and waste, regulate temperature, and of course, protect against disease. Moving on, let's compare exocrine and endocrine glands. Exocrine glands secrete products through ducts like sweat glands do. Meanwhile, endocrine glands release hormones directly into the bloodstream, such as the thyroid gland. Alright, now let's break down eccrine or merocrine, apocrine, and holocrine glands. Eccrine or merocrine glands secrete via exocytosis. Think sweat glands. Apocrine glands release part of the cell along with the secretion, like mammary glands. And holocrine glands? The entire cell disintegrates to release its product, as seen in sebaceous glands.